Good morning, everybody. Thanks for the invitation to give this seminar, this keynote seminar at the Congress. I'm very glad, and I was very glad when I received the invitation from the organizer. So I hope we can have less, less than 1 hour together, and let's see overview what actually are interaction and relationships between fungi and algae, and what we can find in the lichen symbiosis area. Let's say, I decided to take care of this topic because. I was thinking about the use of these two words, interaction and relationships, when we talk about the symbionts in lichens. Interactions An interaction is something that concerns communication between two entities. But when this communication starts to be more deeper, let's say, and when the two entities become connected, they install a certain relationships, and things start to be related, almost interdependent between each other. Because an interaction develops into relationships, we need time and we need intensity. During the time, in fact, the compatibility between the two entities increases and increases till the point that the two entities, let's call the two partners, starts to adapt to each other and even may start to co-evolve together. This co-evolution and co-growing together leads to the formation and to the building of certain structures to serve and to support certain functions. So the two partners start to optimize their life optimize their life in order to better fit to a specific environment. In fungi, this happens because this is majorly led by the eco-nutritional mode of the fungal mycelium. In fact, fungi can build dynamic interactions with their host, so with the other partners, basically in order to serve their requirements to feed. So different nutritional lifestyles in fungi can also switch between one and the other, and fungi can be biotroph, necrotroph, saprotroph. But when the interaction between the fungus and the host starts to be less dynamic and starts to become an obligate relationships, and you see there is an interdependence of one partner to the other, or both partners to each other, then a certain stability is reached, and we can identify two main key relationships that can be the parasitic one or the mutualistic symbiotic one. And these kind of relationships are also identified as association, are typical in the formation of a dependence between a host and the organism which is hosted. Parasitic and mutualistic symbiotic relationships in the fungal kingdom in the fungal tree of life are very frequently recovered to be interrelated. In fact, we have lineages, fungal lineages that share different lifestyles, and in Ascomycetes especially, there are lineages which can host taxa which have developed very different lifestyles and share different evolutionary adaptation. The most key relationships we said that are the parasitic one, and there are those involving plant pathogens, for example, or fungi which can parasite other fungi, or even fungi which can parasite lichens, the well-known lichenicolous fungi. Alternatively, symbiotic relationships are the typical mycorrhiza symbiosis and the lichen symbiosis. And lichen symbiosis is best identified by the multiplicity of thallus forms that a fungus can build when co-growing with a certain algal partners. But there are also some relationships that are transient, that are symbiotic, but they are transient. This means that they are not so stable. Their stability can be broken, and these are observed between usually melanized fungi that are that can co-grow inside the plant bodies, and these are the fungal endophytes. But the same can happen with fungi that can co-grow inside the lichen talus, so the endolichenic fungi. And finally, we can also detect in nature very primitive forms of lichenization of these melanized fungi growing together with algae. And exactly from these, let's say, transient symbiotic relationships, we can start to trace an evolutionary path that leads practically from the fungal-algal interaction to the most developed fungal-algal relationships exemplified by lichens. In fact, in nature, we observe fungi and algae growing together, having the potentiality to interact, but whether there is already an interaction or an exchange of nutrients has not been proved yet. And then we have forms in which the fungus starts to develop together with the algae. This happens usually in very oligotrophic environments, on on bare substrates, usually rocks. And in this case, the fungus interacts with the algae, but whether it has established a relationships, a true relationship, has not. But it has not established a complete relationship so far. And then when fungi lose the melanized algae, so it seems they start to contact algae in a more specific way, and they start to enwrap algae in a matrix, and these fungi are those that can optionally lichenize, choosing between the lichenizing lifestyles and the saprotrophic one. And in this case, we observed specific interactions between the fungus and the algae, and in only few cases, the development of a certain relationships. But finally, the best exemplified relationships are those represented by lichen symbiosis, in which there is a specificity between the two partners, so between the fungus and the algae, and there is not only interaction, but there is a true relationships, because in some cases the symbiosis does not develop if exactly those two partners are not co-present in nature and do not join each other to build the lichen talus. So at the base of our, let's say, called this evolutionary ladder, we have black fungi that grow very frequently in nature together with algae. These melanized fungal hyphae usually spread and branches among algal colonies, and they can be found in very peculiar environments. They are usually characterized by extreme conditions, oligotrophic growth conditions. One recently discovered examples come from the Atacama Desert, and has in focus two very extremophilic organisms. One fungus, that is Orte vernecki, and the other, the photobiont, the algal genus Tunaliella. This couple, Orte vernecki together with the Dunaliella, so-called Atacamensis, have been discovered to grow on spider webs at the entrance of a cave in the Atacama Desert. Interestingly, the fungus and the algae grow together on the spider webs, and they form clumps that remain attached to the webs of the spider. The more interesting part is that these two organisms are both extreme halotolerant cells. One is a yeast, Orte vernecki is a black yeast, while Dunaliella is usually the dominant algae in hypersaline environments. And both of them have become models to study salt adaptation in very in very dry and extreme environments. Interestingly, in this consortium of Horta and Dunaliella, we may have also some bacteria, but when we grow them on cultures and we try to separate them and then regrow together in order to study whether the two interact and whether the fungus uses the photosynthesis produced by the algae as nutrition sources, we failed in co-growing them together. In fact, we developed multiple cultural experiments in order to let the fungus and the algae growing first axenically and then together. But we did not succeed in obtaining colonies of the green algal Dunaliella, while only the fungus could grow. So it seems that something is missing in this association. Either the conditions in vitro do not reflect those that are in nature, and so we do not have the same parallelism with nature, or some microorganisms may still play a role in this symbiotic in this association, let's say. But when we move to rock substrates and we move towards higher altitude and drier environments like mountain desert and so on, we find that there are on rocks lichen-like association between filamentous black fungi or also microcolonial black fungi and algae. In these cases, again, fungal hyphae or cell clumps of the melanized fungi grow tightly with the algal cells. And these algae can belong to different genera. We both have Trebuxia, but we have also Cocomixa. So we find a certain diversity in this association. These two organisms usually develops inside very thin rock crevices where a little bit of humidity is kept, and they are recurrently found together. These two fungi usually belong to the genera Lichenothellia and Saxomyces. They have been characterized as such and in the order Lichenothelielis. They have both the characteristic to grow with the algae in nature, and they can be isolated in cultures. They can be axenically isolated in cultures, and we were interested to see whether their growth in culture is potentially influenced by the interaction with the algae. So if the fungus uses the photosynthesis product of the algae, and if the presence of the algae influence and maybe speed up the growth rate of the fungus, and whether the fungus and the algae growing together may build more organized structures that recall in a way a lichen thallus. We saw that actually there is not a substantial variation in the growth rate of the fungus in the presence with the algae, and we observed that actually when the two grow together, they do not develop through lichen-like structures. Instead, the algae grow intermixed between the hyphae, and the hyphae just grow. So it seems that there is not significant interaction between the fungus Lichenothellia in this case and the algae, and and we had no hints to the formation of a lichen-like thallus structure. This is likely due to the melanization of the cells. In fact, many discussions we had in these past years thinking how to bring together these two organisms were centered on the problem of the melanization of the cell walls, and also together with Martin, we often thought that melanization hindered the fungal-algal association in a proper way. In fact, we see that when fungi lose melanization in their cell walls, there is a more tight contact with the algae, and the fungi starts to enwrap the algae. This happens in a fungus that can optionally lichenize, and it became a model since already more than 15 years ago. It was discovered by Mats Vedin, and then it was the focus of our researches because the fungus can grow as a protrophic on dead wood or in a lichenized, in a lichen-like form on bark. And we saw that actually when the fungus and the algae are isolated and then put together in co-growth, at this stage, the fungus develops its mycelium above the algal colony, almost forming like a sheet of hyphae, and the fungus starts to contact the algal cells and wrapping them, forming a filamentous matrix over them, meaning that there is a symbiotic association or a hint to a symbiotic relationship. And this happens mainly when the fungus grows together with the algal genus Cucumixa, which is the algae that is isolated from the environmental samples on bark. Definitely in this fungal-algal association of Schizoscylon, Albacin and Cucumixa, there are actually third players that are bacteria, and bacteria have also investigated in the past years, and we saw that actually they are present and they are closely associated or located next to the algal colonies, but whether they influence or interact in a way with the algal cells or the fungus was not proved. We can remaining on the melanized organism. Even though we do not see the formation of lichen-like structures, just co-culturing the two organisms together, we can even force to co-grow fungi and algae using alginate inclusion. And we attempted this a few years ago when we put together the black fungi and the algae in the same alginate inclusion, and we let them grow. The colonies were growing in and out of the alginate inclusion, and were developing in a very nice and a very clear way. Still, in the case of the melanized fungus, we saw only just co-growth of the fungus and the algae, although it was to be really careful to interpret this intermixed growth as an interactions, a proper interactions. Definitely, the fungus profit from the growth together with the algae because it was not damaged during the growth. But more interestingly, when a not melanized fungus is growing with a lichen photobiont, then we see that actually there is contact of the hyphae with the algal cells. So this experiment helped us to, let's say, start new approach in the co-growing of two organisms that in nature live together with algae, but have not observed for any peculiar morphological structures. But what it happens that we have cases in nature that were also melanized fungi can grow and form true symbiosis with true lichen symbiosis with algae. And this is the case of the two genera, Chistocollus and Rhacodium. They were studied many years ago by me and by other colleagues, and they are very peculiar because in this symbiosis the fungus develop its hyphae along the filaments of Trentifolia. Trentifolia is a filamentous green algae, and the fungus encloses completely the filaments of the algae. The algae is the central axis of the lichen thallus. It is the one giving, let's say, shaping the thallus, the lichen thallus. But interestingly, this is happened with these two filamentous melanized fungi. And so there is a lichenization and there is a formation of a true relationships in this group of lichens. Still, it remains peculiar because Cistocollus and Rhacodium are not fungi from the order Lecanorales, from the Lecanoromycete and the class Lecanoromycete, where the majority of lichenized fungi belong, but they belong to Dotidiomycetes, and they belong to an order in which we have many other fungi with many other different lifestyles. So the order is the order Capnodiales, in which the Cistocollus and Rhacodium are individually monophyletic, and they both have evolved the possibility to grow as lichen. But definitely the best relationships we can see in nature are those exemplified by the lichen symbiosis. Lichen symbiosis have nowadays revised in their definition. In fact, David Oxworth and Martin Grube just last year reformulated the definition of lichens as a self-sustaining ecosystem formed by the interactions of exhabitant fungus and an extracellular arrangement of one or more photosynthetic partners. In these structures that the extracellular fungus and the photobionts form, we have an uncountable number of other microorganisms that can be other microfungi, microalgae, and bacteria. These additional participants to the lichen symbiosis may be and may not be isolated in arsenic conditions, so they can in some cases be grown in cultures. In other cases, we are unable still to isolate them. They can shape the phenotype of the symbiotic of the symbiosis itself, but still for the majority of them has not been demonstrated which is the exact role. Lichen symbiosis was viewed under a model of adaptation evolution considering the two main partners, and this was the fungus, so the mycobiont, and the algal partner, the photobiont. And under let's say an ecological climate change event, the lichen itself undergoes selective pressures, and in this process of adaptation, the fungus may be able to switch the photobiont partner and searching for a photobiont which is better adapted to the environment under changing. In this case, the new relationship that is established may develop into a really new lichen species. So the two partners, the new couple of the two partners evolved and becomes a new lichen species. But if we consider all the other organisms in lichens, what do we see? Well, we see that there are many algae, and this is true. So the switch between one photobiont to the other is possible, and it was observed that within the lichen thalli we can have different population of photobionts that have different ecological performances that can be characterized morphologically and by their ultrastructural characters when they are analyzed in cultures. And we saw that in the particular case of Trebuchia, we may have many different Trebuchia in a lichen thallus. Trebuchia has been, let's say, reordered in the past years by a group of us. Many of us took care of revising the phylogeny of Trebuchia, and the original 4 main lineages in Trebuchia have been confirmed and have been even in detail analyzed better. Inside the lichen thallus, we can have representatives also of different lineages of Trebuchia, not only of taxa belonging to the same lineage. But we have also many other fungi in lichens, and these fungi can be symptomatically present on the thalli, and they usually undergo, they are usually recognized as the lichenicolous fungi, but they can be also asymptomatic, and they inhabit the lichen thalli, and they can be detected only when they are isolated in cultures. These have been started, let's say, already in 2009 by Betsy Arnold, but then Betsy, her group, and we succeeded in isolating many different endolichenic fungi. And we see that according to the origin of the lichen thalli, we have also different composition of the endolichenic fungi. And culture isolates demonstrate this. Not only we have nowadays also the possibility to sequence with deep sequencing the whole diversity in the lichen thallus. So metabarcoding analysis helped to identify the whole fungal community, the so-called lichen microbiome inside the individual thalli. And it was possible to distinguish three major fungal fractions that are representative of the different group of fungi more or less specifically associated to the lichen thalli. In fact, one fraction is represented by the lichenicolous fungi that are usually species-specific to the lichen thalli, while the other two fractions are fungi that may be present in the lichen and using the lichen as a springboard to adapt to other niches, and others can be just transiently present. They are randomly present in the thalli. They are occasionally found here and there, and probably their presence in the thalli is not influencing the whole thallus phenotype. So if we consider the lichen thallus as the holobiont in itself, in this case, adaptation and evolution do not regard only the fungal partners and the algal partner, but also the whole complexity of a microorganism inside the thalli. So under selective pressure and and under different ecological conditions, not only the algal partners may switch and may be selected for a better one, but also we have the composition of the fungi that may change, as well as those of bacteria. But in the bacteria part, I did not go in the detail. So in this case, the adaptation and evolution of the lichens itself includes also variation in the composition of all its partners hosted inside. So, and this is actually where we are now. We are just at the point where we want to understand how ecological variation shape the diversity of the lichen microbiome and the lichen associated algae. In this respect, I personally have developed, and I'm leading two projects in which we seek to understand if the lichen microbiome is stable and how much it varies according to the different ecological conditions and according to the diversity of the lichen species. So is the microbiome more related to the ecological parameters or more related to, or is more species specific to the microbiome, to the principal microbiome? And secondly, this ecological study takes in consideration also whether we have a niche differentiation, niche diversification of the lichen microbiome in extreme environments, and this includes the comparison of cosmopolitan lichen and their microbiome, also taking in consideration species from the Antarctic regions. And finally, the diversity is one point, but the presence of metabolites, exudates produced by all the symbionts in the lichen thalli is the next, let's say, long-term goals that I think we all aim to reach and to understand. In fact, this big and huge, I would say, organismal diversity that we have in the lichen is certainly responsible for the expression and production of different secondary metabolite profiles. And we are now seeking this study together with the group of Innsbruck that is actually leading the research. And we took as a model system, as a reference system, the lichens Tephromilatrax, Xanthoria paretina, and Cladonia gray. And in these symbiosis, we search for metabolites produced by the algae, by the microbionts. We co-grow them in cultures, and we analyze what they produce and they release, and whether these products, metabolic products, may be used by either one or the other partners. So with this, I finished my seminar. I hope you found it more or less interesting. It was like a review of what has been done in the past years, and definitely this is the product of a big and long-standing research collaboration with all these people. Some have been closer to me, other less, but I think in these big families of lichenologists, we have established good relationships to work further and to investigate lichen symbiosis under different aspects. So thank you to everybody, and I'm happy to take questions or to discuss about any topic.